Thank you for a very kind introduction. Good afternoon, everyone. First of all, I would like to thank organizers for giving me this opportunity to talk about our work. My name is Natthakan Thongon. I'm from MD Anderson, and I'm working with Dr. Simona Kohler. Our work was aimed to study the telomere shortening because it is a primary molecular cause of DNA damage during the human aging. Today, I'm talking about how we use flow cytometry to dissect the mechanism of hematopoietic stem cell exhaustion induced by telomere shortening. Telomeres, Telomeres are physical ends of the chromosome, and they become shorter and shorter every time that the cell divides. Both stem cell and somatic cell go through the replication process, but during the replication, the stem cell undergo very less number of replication. And the most important thing is the stem cell express telomerase, which is the enzyme that prevents the chromosome shortening. For this reason, telomere of the stem cell get shorter later on. On the contrary, somatic cells do not express telomerase. They undergo multiple rounds of replication. Their telomere get shorter very fast, and at some point, they become too short, so the cell can no longer replicate. And eventually, they become senescent. However, both in stem cell and somatic cell, when the telomere is shortened, the cell initiate the DNA damage response with activate mechanism of apoptosis or senescence through the activation of P53 or P16. And these mechanism of actions induce the functional decline of stem cell, which resulting in aging. Here we want to understand how telomere shortening affect hematopoietic stem cell functions. To do so, we use a telomerase deficient mouse model, which is a mouse model which has no telomerase. So in general, the telomere of the mice are very long. So in order to obtain a short telomere mouse model, we do need to cause the mice for 5 or 6 generations. We started causing the mice from generation 0 with the telomere still intact. So telomere of mice in G0, they are still normal. And over the coursing of the mice over the time lead to their progressive telomere erosion. So the telomere of mice in the generation 5 and 6, they become clinically short and remain uncapped. This induce their DNA damage response, which resulting in their severe tissue degeneration and a phenotype of aging. Our goal is to understand how telomere shortening affect function of hematopoietic stem cell. To this end, we analyze their hematopoietic stem cell compartment in the mice with short telomere, G5 or G6, as compared to the mice with long telomere, G0. Before going to the experiment, I would like to give you a brief overview of mouse hematopoiesis, which is a very complex system. So over the year, the stem cell and progenitor cell are recognized by the different surface marker as shown here in this slide. Briefly, the hematopoietic stem cell is based on the top of the stem cell. Based on the top, which produce the multi% progenitor cell or MPP1, which is metabolically active. That gives rise to MPP2, 3, and 4. And later on, those cells, they are produce the different type of mature effector cell in the blood, including platelet, erythrocyte, neutrophil, monocyte, T cell, or B cell. Over the years, it has been shown that multi% progenitor cells have the different lineage potential, which is very important. For example, MPP2 produce megakaryocyte, erythrocyte. MPP3 produce granulocyte and myeloid cell. And MPP4 produce lymphoid cell, and this is in the condition of homeostasis. Sorry. But in stress condition induced by DNA damage, inflammation, or transplantation, the stem cell produce more MPP2 and MPP3 population. That means they produce more megakaryocyte, erythrocyte, granulocyte, and also myeloid cell. And here we wanted to understand what was happening with the stem cell in the setting of telomere shortening. To do so, we used flow cytometry analysis and sorting of highly. You cannot see the title here. To sort the highly purified hematopoietic stem cell population. And in this table, we show the markers of our, we show the marker. Perfect, thank you. In this table, we show all the markers and gating strategy that we use for flow cytometry to define the different stem cell population. As shown here, we use 710 for cell viability, and from large single cell, we selected only linear negative population. And from here, the positive selection of LSK, which is our stem cell compartment, is based on the expression of scale 1 and c-KIT. And from this population, together with double negative CD135 and 34, and also negative for CD48 but positive for CD150, we can recognize our HSC or hematopoietic stem cell population. And from negative CD135 but positive CD34, we can recognize MPP1, 2, and 3 population according to this gating. Lastly, from double positive of CD135 and CD34, together with CD48, we can recognize the MPP4 population. And based on this panel, we observed that telomere shortening affects hematopoietic stem cell population. We saw that we saw the decrease of HSC both inside the stem cell compartment, LSK, as well as inside the bone marrow of the mice with short telomere, G5, G6, as compared to the mice with long telomere, G0. And from this experiment, we observed decrease in the number of HSC, increase the number of MPP2, and decrease the number of MPP4. Our question was to understand if the loss of HSC was due to apoptosis, senescence, or autophagy, and the increased number of MPP2 and decreased number of MPP4 was due to aberrant differentiation of the stem cell. To do so, we used flow cytometry strategy, both BD influx and LSR Fortesa. In terms of the sorting, we use this following marker combination to sort the hematopoietic stem cell to perform single-cell RNA sequencing, single-cell ATAC6 sequencing, as well as the bone marrow transplantation experiment. And in terms of the analysis, we use this following the marker combination to analyze apoptosis, senescence, and autophagy. And we demonstrated that the telomere dysfunctional HSC do not undergo apoptosis, senescence, or autophagy because we didn't observe the difference of annexin 5 staining for apoptosis, cytol ID lyso for autophagy, and the LEC analysis for senescent cell in the stem cell from with short telomere G5, G6 as compared to stem cell from long telomere G0. And next, our question was to understand why the HSC was decreased and MPP2 population was increased. We used the flow cytometry strategy as described above here, which is the same marker as I described earlier, to sort the hematopoietic stem cell. And it was an extremely difficult experiment because we do need to combine at least 4 mice in order to get something like 2,000 cells of their stem cell to perform single-cell RNA sequencing analysis. And from the analysis, we recognize the different cluster of the cell. That means cell in each cluster different from each other based on their gene expression profile of the stem cell. And from here, we understood that in cluster 0, as I show here, the cells were in the lower differentiation state, or in another word, they were more quiescent. As you can see from these 3 genes, which are the marker of quiescence, which were highly expressed in the cluster 0. Together with the globally, when we performed the analysis of cluster distribution, we observed that the stem cells from short telomere G5 and G6 were highly active. And we observed the almost completely loss of the cluster 0 in the mice with short telomere G5, G6 as compared to the G0. To confirm this result, we use these following for cytometry setting to analyze their cell cycle state inside their stem cell population. And we analyzed based on the expression of Ki67 and the DAB as their DNA markers. Our result demonstrated that their stem cell from short telomere from G5, G6 were still under cell cycle progression, which means they are still active. And below is showing how we set up the flow cytometry. And we also sorted their hematopoietic stem cell to perform single-cell ATAC sequencing. And this single-cell ATAC sequencing analysis allow to understand how the stem cells are going to differentiate. And from the result, we observed that their stem cell with short telomere from G5 and G6 are prone to differentiate into megakaryocytes, which explains why we observed their MPP2 increase in MPP2 population, as I showed earlier. And moreover, we also see that their transcription factor that active are also inside the interferon family. Similar result was observed when we analyzed the different gene expression profile between G0 and G5, G6 by single-cell RNA sequencing analysis. And we observed that there was an activation of cytosolic sensor of pathogen-associated DNA, which is known to activate the interferon signaling pathway. So our question was why the cytosolic sensor of pathogen-associated DNA was activated. And is it possible that their cytosolic DNA due to their telomere degradation was increased? To respond to this question, we used Amnis image stream machine and together with this flow cytometry strategy to detect their cytosolic DNA inside the stem cell. And our results show that their stem cell with short telomere have an increase the amount of cytosolic DNA as shown here. In conclusion, we use flow cytometry analysis and sorting strategy to dissect the functions of highly purified hematopoietic stem cell populations. And we demonstrated that their telomere shortening induced hematopoietic stem cell activation and differentiation toward the megakaryocyte due to the activation of interferon response, which is induced by the release of cytosolic DNA. And you can read, if you are interested, you can read our full paper in Nature Communication. And I would like to thank my PI, Dr. Simona Kolar, and everyone in our lab. And special thanks to everyone from flow cytometry and the South Campus. All the work and the paper could not be possible without help of you guys. And thank you very much. We really appreciate your help. Thank you.
Okay, do we have any questions from the audience? No questions. So maybe you can also stay on after the whole session if there is more questions.
Sure.
We can continue.
I cannot hear. Can you repeat again?
The progenitors could differentiate, or there is a stop in this level. And if they differentiate, the final cell, for example, the megakaryocytes, are functional or not?
So one is differentiate. I mean, I do not know it. We didn't go to further this part. What, in my perspective, I think they are actually.
There's one online. Mikey's asking, were you able to quantify or measure telomere length by flow?
Yes, we used their technique called FlowTex. Yeah, and that's it. The principle is like there is the fluorescent probe that's recognize their telomere repeat, the latest base pair. So, and yeah, we use for cytometry to analyze that. So the higher the signal can tell us the longest of the telomere.
Okay, thank you.
Thank you very much.
Okay, our next speaker is Cheng-Yen Chang. She got her PhD just a few months ago from Baylor and is going to talk about her studies during the PhD. And specifically her topic today is chronic exposure to ultrafine carbon black reprograms macrophage metabolism and accelerates lung cancer. You're welcome.
Hello, everyone. Thanks for the introduction and thanks for the opportunity to present in this meeting. Today, I'm going to share our work about a small size particle called carbon black. By its nature, this particle is black, so it can block the fluorescent signal. So I would like to share how we overcome this difficulty and analyze the role of this particle in lung immunity. So as you can see here, healthy human lung looks pretty white, but tobacco smoker's lung is really black. In 2015, we identified the material in the smoker's lung. This is called nano-sized carbon black. This is a reason that caused the blackening of lungs. Single molecule size is 15 nanometer, but they tend to form aggregates around 50 to 75 nanometer. So they tend to form the aggregates. Because the size is really, really small, so the particle can actually penetrate deep in the lung and retain the lungs long after the exposure. We developed a carbon black mouse model as shown right here, carbon black lung versus PBS lung. Carbon black we found can actually induce emphysema. This is the disease that can cause shortness of breath. And because carbon black can induce protease expression, so they can degrade extracellular matrix. You see the alveoli right here, it's pretty large compared to the BBS one. So overall, decrease the total lung surface area, so make gas exchange become very difficult. Carbon black not just being found in tobacco smoke, but also found in wildfire and carbon black production factory. So in fact, any kinds of organic matter undergo incomplete combustion can generate these carbon black particles. And the particles just get into the air, and everyone can have a chance to expose to this particle. So I want to point out a very special immune cells right here. This is called Aveolar Macrophages. As you can see, this is the macrophage isolated from the emphysema patient, and this patient is a heavy smoker. You can see carbon black particles not just in the cytoplasm, but also into the nucleus. That means the carbon black particles can actually cause DNA damage. So if you look closer into the cytoplasm of alveolar max, this is PBS and this is carbon black. You see chunks of carbon black aggregates inside the cytoplasm. And some of the particles can actually get into the mitochondria, cause the structural damage. So we think this can decrease mitochondrial respiration, which is true because when we isolate the cells and treat the cells with either PBS or carbon black and use seahorse machine to measure oxygen consumption rate, overall, we observe decreased oxidative phosphorylation. We also measure lactate in the cells. Carbon black treated one showed increased lactate. And by the way, lactate is the byproduct of glycolysis. As expected, glycolysis increase in these carbon black treated cells, as shown right here, increased extracellular acidification rate. Next, we want to know what's the immune profile in these carbon black lungs. As mentioned before, carbon black aggregates inside the cytoplasm. So we cannot use the regular flow cytometry, the fluorescent-based flow cytometry to measure the signal because it basically just blocks the signal as shown right here. Alternatively, we use cytometry by time of flight or mass cytometry to detect the signal. Different from the conventional one, mass cytometry use elemental conjugated antibody and mass cytometry to detect the signal. The good thing about this is we don't have to do compensation. I think in the morning section, Sarah mentioned how to analyze automated or computational data from generated from flow cytometry. Basically, we apply a same analysis. Just briefly, we perform a downsampling and then dimensional reduction and then classification by phenograph and flow sim. We successfully identified 26 myeloid populations in carbon black exposed lung. And in our analysis, we have 4 different groups. Non-tumor or tumor, either with PBS or carbon black. As you can see, the carbon black exposed lungs showed increased infiltration of dendritic cells and macrophages. And these cells, CD103 dendritic cells, CD11B dendritic cells, interstitial MAC, and alveolar MAC, these cells express 11C markers, and I would say 80 to 85% of them are macrophages. Here we show the minimum decision trees, basically using full sim plotting software and the expression of surface markers on the cell to make classification. Again, we have 4 groups here: non-tumor, tumor, PBS, or carbon black. We found emerging populations in carbon black treated lungs. We're curious, what are these cells and what markers they express? And it turns out these are macrophages and dendritic cells express immunosuppressive markers such as PD-L1, PD-L2, and CD206. And most of the cells are interstitial max and alveolar max with double positive PD-L1 and CD206. These cells are considered to be immunosuppressive. So we performed a co-culture experiment. First, we isolated cells and then put them together with splenic CD4 or CD8 T cells using regular fluorescent-based flow cytometry to perform the analysis by using cell trace violet. This is generation 0, 1, 2, 3 to 6. The results shown on the right, as you can see, carbon black is the black one, PBS the blue one. T cells that co-culture with carbon black APCs showed a slower cell proliferation. In most of the cells stuck in generation 2 and generation 3. So based on this result, we found carbon black APCs are immunosuppressive. They can inhibit T cell proliferation. And lastly, we performed a doubly transfer experiment. We isolate either PBS or carbon black APCs from the non-tumor mice and doubly transfer the cells to the tumor model at 2 months of age, and then we sacrifice the mice at 6 months of age. We examined lung tumor and stomach metastasis. We found mucosepia mice that received carbon black APCs showed more lung tumor and stomach metastasis. These is just a histology representative. This is a tumor. This is lymphocytes try to get into the tumor. This is normal looking epithelial cells. This is tumor cells break the basal membrane. So based on this result, we showed carbon black APCs can directly accelerate lung cancer progression. So in summary, we found carbon black can disrupt mitochondrial homeostasis, increase glycolysis. Using mass cytometry, we can identify different kinds of myeloid populations in the carbon black lungs. Also, we found increased infiltration of suppressive type of macrophages and dendritic cells in the lungs. And these carbon black APCs can directly accelerate lung cancer progression. Thank you for listening, and thank you, my lab, and everyone who helped this project, especially the core, the cytometry and cell sort core at Baylor. Thank you, Paul and Joe, for supporting this project and our funding sources. I'm happy to take questions.
All right, do we have any questions from the audience? Any of the online questions filtering in? Oh, here we go. Mike.
It's a really nice talk.
Thank you.
Thank you.
What do you know about the characteristics of the macrophage that you see in those mice with the carbon? Do you have any idea, what cytokines they're making,
Yes.
that sort of thing?
Yeah. This is the macrophage actually show a mix of M1 and M2 phenotype. They, at the very beginning, secrete pro-inflammatory cytokines such as IL-6, IL-1 beta. But once, because this is a long-term model, we expose the carbon black for 1 month. In the following month, we let the mice to take a break, and then we sacrifice the mice for analysis. At that point, we found some macrophages can actually secrete IL-10. So it's more like a balance. Inflammation goes up, and then the anti-inflammatory arms try to suppress the response.
Are there any other questions? Do we have any from online? All right, well, we'll move on. Thank you so much.
Thank you.
Okay, we have the last speaker of the session, Katharina Wohlan. She's a postdoc at Baylor, and she's studying the normal and aberrant hematopoiesis in humans and mice, and is studying the clonal expansion of the hematopoietic cells. So you are welcome.
First, I want to thank the organizers for giving me the opportunity to present our project, which is called Clone Wars, a multiplex mouse model of clonal hematopoiesis. While we age, we acquire multiple mutations in our hematopoietic stem cell population. And most of the times, those mutations do not lead to fitness benefit of those cells. But if mutations occur, in certain genes, it leads to increased self-renewal, increased survival under stress conditions, and also do decreased differentiation, which causes this mutant clone to be the dominant clone in the hematopoietic system. So you can see here on the right that while we age, we accumulate multiple mutations. And the most common ones that are found in clonal hematopoiesis are DNMT3A, which has the highest frequency, and then others are TET2, ASXL1, or P53. A lot of commonly mutated genes in clonal hematopoiesis are also known drivers for hematological malignancies like acute myeloid leukemia, myelodysplastic syndrome, myeloproliferative neoplasm, or lymphomas. If the threshold for their variant allele frequency surpasses over 2%, the risk of developing a hematological malignancy is increased, which is then called clonal hematopoiesis of indeterminate potential, or CHIP. Clonal hematopoiesis is also related to non-hematological malignancies like cardiovascular disease, strokes, and also increased inflammation in general. And this results in an increased mortality risk of 30 to 40%. Apart from aging as a known driver of clonal hematopoiesis, we have other stressors like smoking, obesity, genotoxic stress, inflammation, and infections that cause expansion of certain clones with mutations, for example, in DNMT3A, TET2, or PPM1D, as you can see here. The aim of our study is to establish a mouse model where we can study clonal competition in vivo. For that, we wanted to establish a transplant where we use different mutations to match clonal hematopoiesis that we found in humans, and use genes that are commonly mutated. And after successful establishing of this transplant, we want to establish analyzing tools to study the clonal competition and to monitor changes in the hematopoietic system. So our setup looked as follows. We used 5 different mouse models that harbored mutations in either DNMT3A, TET2, P53, PPM1D, or SMT3. Before we transplanted, we ran a flow for HSCs to determine the cell number of HSCs in the whole bone marrow, to normalize the bone marrow so that we can transplant the same number for each mutant for HSCs, giving them the same starting point. After successful transplant, we collected blood to look at the expansion of those mutant clones over time using digital droplet PCR.
and also stained for lineage markers, meaning B cells, T cells, and myeloid, to follow the development over time in the blood. At the end, apart from collecting blood, we also collected bone marrow to, again, do the dPCR for the whole bone marrow, but also stain for lineage and stem cell markers. And additionally, we sorted for LSK populations and lineage populations to then isolate DNA out of those cells and run the dPCR to look at the clones in those populations. For the sorting, we used amine dye staining to pool mice. So we isolated bone marrow from 3 different mice and stained them either with CFSE, CTV, or CSFE and CTV to then be able to pool the mice before we do c-KIT selection using the AutoMax. The c-KIT positive population we use for LSK sorting, as you can see here, our gating strategy. So we gated for lineage negative and then c-KIT and SCAR positive cells. And then I hope you can appreciate here really nicely that we can distinguish between those 3 mice, sorting LSKs specifically for each single mouse. And for the c-KIT negative population, we use those to sort for lineage cells. Here's an example for the myeloid population. And again, you can see really nicely that we can distinguish between all 3 mice and sort those cells. So here are the results. In the blood, you can see in general that PPM1D and SMT3 mutant cells are engrafting very poorly, and over time you can see them vanishing in the blood. For P53, we have around a variant allele frequency of 5%, which does not change over time. TET2 showed the strongest engraftment at the beginning, which is week 4, and then you can see it's doubling at week 16 and then stays stable until the end. And for DNMT3A, at the beginning, we see slow engraftment, but then with time, as you can see here in the green color, it's going up to 15%. When we compare bone marrow, blood, and the progenitor and stem cell population, we can see in general that TET2 is the dominant clone in all of those compartments, and followed by DNMT3A, and then P53. And again, as we already saw in the blood, PPM1D and SMT3 are almost absent by the end of the experiment. Next, we looked at the bone marrow cells that we sorted for lineage markers. And as you can see here, for the B cell and myeloid population, again, TET2 is the biggest fraction. And especially in the myeloid population, it's really interesting that TET2 almost takes over and suppresses all the other mutant clones. So then we looked at the lineage population in the blood over time. As you can see here, in general, we don't see any changes for the distribution of the different lineage populations. But using our system that we have CD45.1 wild type cells, and the mutant cells are either CD45.2 or CD45.12, we are able to distinguish those cells in the lineage populations. And as you can see here, at week 4, we have like an even distribution, being 50% wild type, 50% mutant in all of the lineage populations. And then over time, you can see that the mutation, that the mutated clone population increases and completely takes over in the B cells, myeloid, and T cells. In summary, we were able to successfully establish a multiplex mouse model to mimic clonal hematopoiesis in the mice. And in general, what we saw under just normal conditions is that TET2 seems to be the dominant clone here, followed by DNMT3A and P53. And in the blood for the lineage population, we saw that it's that the mutant clones take over time. So our future plans for this study is to study the clonal competition after adding the stressors that are known for clonal hematopoiesis, like inflammation, smoking, or cytotoxic stress. And those experiments are currently undergoing. With this, I want to thank everyone that contributed to this project. It's a huge effort between 4 labs contributing to this study. And with that, I want to thank for your attention, and I'm happy to take questions.
Do we have any questions for our emerging speaker, scientist?
Hi, really nice talk. I was wondering why you picked CFSE as a dye, and did you try also other proliferation dyes?
No, we started with using CFSE and CTV because we can like nicely distinguish it in flow. Then combining it with all our other markers, making it the best combination for staining for flow.
Was it the best combination because you knew CFSE fitted in your panel?
Yes.
Okay. Thank you.
All right. Do we have any other questions? Do we have anything from our online participants? All right. Well, thank you so much. Great talk. So we are scheduled for a 15-minute break right now. Before y'all disappear, do remember that if you parked in the next door garage for the UT, we do have parking reimbursement slips, so you can pick one up by showing your badge to our desk, or come up and talk to one of us, and we can get you taken care of. We're gonna take, like I said, a short break, and be back here for our next section. And then later on, after that, we do have an ice cream social where, unfortunately, our online participants can't partake, but we all can have a little Amy's ice cream. Please be back at 2 o'clock.
Hello. There we go. So welcome everyone to the last session of the day. This is the session of sorting into multi-omics session. Yes, I am muted. Thank you. So it's my pleasure to present Tricia Rogers. She is the Associate Director of the flow cytometry facility at the Broad Institute of MIT and Harvard, and she's worked in the field for flow cytometry for over 16 years. So Tricia, put together the flow cytometry facility there at the Broad from scratch, and she's been driving this facility for the past 6 years, which supports around 400 researchers from over 30 groups and programs. And the facility has grown rapidly and now includes over 20 flow cytometers, genomics, and imaging instrumentation. And on a personal note, so I've known Tricia for quite some time, and we've been friends for many years, and we've collaborated together. And I have to say that she was always a person that inspired me a lot. She really knows what she's doing, and the proof is how she's been driving the facility at the Broad and starting it from scratch. And actually, she was one of the first persons, I think, that I knew that started thinking of putting together genomics into a flow cytometry core facility. And in fact, while she was putting together the lab, she was also finishing her bachelor's, and she was heavily dedicating herself to genomics and already thinking ahead, way ahead of most of us. So this is what she's going to present today, and I'm looking forward to hearing.
Thank you, Rui, for that introduction. And yes, side note, I was a Sooner. I'm a native Oklahoman, but I didn't really belong. I wasn't really a Sooner. So no rivalry here. I dropped out and moved to Massachusetts in 2004, and I finished my degree just a few years ago, actually, at UMass Boston. So now I'm a Beacon. So yeah, today, so thank you to the organizing committee for inviting me all the way down here. This is actually my first meeting since 2020, where I was at AGBT and Marco Island, which was just rife with COVID. And so I'm really excited to be in public again. So what I'm going to talk about today is just, how do you take your flow cytometry lab and move towards genomics? A lot of us are interested in doing this because it's a very popular assay, obviously. We're all doing single cell genomics in our labs now. And so I'm just going to show you how I did it, and maybe you can have some takeaways so you can do this in your own course. So the overview of my talk today is just going to give you a story of basically how Broad Flow Cytometry Facility was created. As Rui said, we're fairly new. We've only been around since 2016. And then I'm going to talk about how I transitioned to a flow genomics lab. I don't officially have that title for my lab yet, although it should be coming. Like, people get so confused when they're new at Broad, and everyone tells them, oh yeah, go do 10x in the flow lab. And they're like, what? That's not typically where you go do 10x work. And then I'll briefly touch on future expansion. So what am I thinking about in the next few years of adding to the facility now that we already have kind of a good pipeline for flow to genomics? So let's get started about, just how we started flow at Broad. So a little overview of the Broad Institute. It's Broad, not Broad, like everyone wants to say. We have 3 different kind of groups at Broad. So we have our scientific programs, we have platforms where you can kind of think of them as core facilities, but they're really more collaborative. So they really are more involved in your project than at just a service facility. And then we have recharge centers, and that's kind of where I fit in, just based on the structure of how I charge the end users. And so a lot of what we do revolves around human disease and research. And so we have programs like the cancer program, and within the cancer program, we have drug discovery and chemical biology and therapeutic science, so CBTS and CDOT. And so they do a lot of high throughput screening and testing of compounds of interest. And then we have cell circuits and epigenomics. And so they do a lot of the single cell work currently with the Klarman Cell Observatory at Broad. And then the platforms, we're all well aware of, like the imaging platform with Ann Carpenter and Cell Profiler. That's a big part. Side note, she has no actual imaging equipment, though. A lot of people get confused by that. And then we have the genetic perturbation platform, which does a lot of CRISPR screening, and then genomics platform, which is also, world-renowned. We have hundreds of sequencers, and it's really been crucial to pivoting to doing sequencing of the new COVID variants and testing for the state of Massachusetts for COVID. So we are not very big. We're only a few buildings, but it seems like we're a lot bigger than what we are because we collaborate with almost everybody. So this is the flow cytometry facility. So we were founded in 2016 from an internal Broad Next 10 grant. So basically back in 2014, 2013, the Institute was looking at the next 10 years of the Institute and how they were going to evolve and meet the needs of the researchers and the science. And so a small group within Broad decided to put together a proposal to form a flow cytometry facility because there was not one at Broad prior to me joining. So the researchers had to go all over Boston in order to do flow. A lot of them have appointments at their home institutes like MIT, Harvard, Dana-Farber. And so that's where they were doing flow cytometry. But you're not really given preferential treatment if your lab isn't located there sometimes. And so it was really important to bring that back to Broad so researchers could have on-demand flow cytometry. And I had worked a little bit with Broad while I was at Harvard previously, working in a core there with the Reprograms lab. And I knew a little bit of how they worked, which was all the time. Like they took no breaks. They worked very closely with the hospitals, and they did a lot of single cell work on tumors coming directly from patients, and they were coming over to Harvard. And these were the early days of single cell work where we sorted like 5 cells and we were excited into plates to do single cell RNA-seq on them. And they would often show up at 7 or 8 o'clock at night. And so I was like, yeah, I'm not going to do this when I go to Broad. And so I had to think about how to set up a facility from scratch to really serve the research community there. And the one thing I came up with was making it truly a self-run facility. So I gave the reins basically to the researchers so they could run anytime they needed, so they didn't lose those precious samples. And so since I had to buy everything from scratch and design this from the ground up, I could really go in and choose the instrumentation that would fit this model. And 2015 was a really good year for this because there was so much new stuff in the market at that time. And so I didn't have to go with kind of the standard, the status quo of instrumentation. And so I could bring in things like the Sony SH800s, which are very user-friendly and easy to train on, which was key for us to get going. And so originally we were only supposed to buy 4 flow cytometers in that original proposal, but instead I bought 3 cell sorters and then 4, I don't need to lose track, maybe 5 cell analyzers. But we basically doubled what we thought we were going to bring in, which was great. So it gave a lot of flexibility and early access to all of the users who were really, craving this at the Broad. And so as you can see in a few slides, we currently have about 45 instruments in the facility. It has grown rapidly since 2016. And so if we break down what we have currently, so this is a picture of the other, basically our lab is split in half. It's all within one room, but the front half is all flow cytometry and the back half is now turning into a mini genomics core. But we're still mostly flow cytometry. All the genomics is catching up pretty quickly, and that's largely because genomics equipment, they come out with new stuff every year. It seems like flow cytometry will have these spurts and growths, and genomics is constant. So there's high turnover there. Sample prep is another big area that we're trying to expand in because there's a lot of protocols out there, single cell attack, multiome from 10x that requires a lot of diligent sample prep. And so this is equipment that we're actively going out there and investigating and trying to bring into the facility. And then imaging, and I can talk about that a little bit later, but spatial transcriptomics is the next big thing. And so that's the areas that we're kind of going into. And so we have a team of 7 full-time staff, and we're, like I said, all within one lab. And we have a range of expertise from flow cytometry to molecular biology. So I actually have a molecular biologist in my group. He's a staff scientist. And we partner with our genomics core because I don't want to go around them. I don't want to compete with them. I want to complement them. So I want to work alongside my genomics core. And actually they are in my lab. And so this is kind of what we have right now. It's a lot of stuff. I'm not going to go in details about what we have, but we have a lot. So we have, like Marie said, about 20 flow cytometers now of every make and model. We have a lot of molecular biology equipment, so we're really a 10x heavy lab. We're always assessing new technology, but at the end of the day, we want to buy stuff that actually gets used by the researchers, so we buy what they're using. We are getting in pretty deep into the sample prep equipment, so our newest acquisition there was the Levitas Bio Levicell, which I can talk more about offline. And then you can see that I have a cryostat and a microtome as well. So those are kind of our newest purchases. And then the imaging is, we're actively looking at some Leica microscopes that have just launched. So we're adding to that area as well. And so I just like this picture because it highlights just how dense the lab is right now. Because when I took this picture in 2017, I think this was for Cyto Boston when I did a single cell genomics workshop. I was really proud of this picture from 2017. I was like, look at all this cool stuff. And then I literally have no space left in my lab. Every month we're trying to move things out to move things in. We're putting things that we're not using as often on shelves to store them. And so it's rapidly evolving in our lab. The asterisk is still there though. It's getting tighter and tighter. You can see that bench keeps moving in. And I'm like trying to push how much further it can go in legally without getting yelled at by facilities. And so really our mantra is adapting to the needs of the Institute. So, it's really getting outside of our comfort zones to help drive the science forward. And so just because I don't know how to do it doesn't mean I shouldn't bring it into my lab and figure it out. That's kind of what we're there for. We're there to help and we're there to push science forward. So we're always testing new equipment. We're trying to sign up to be early adopters and test beta instruments. And I'm adding expertise to the group that can help with the weird stuff because I don't know how to do everything. I'm not a molecular biologist, although I'm learning about all of this stuff. So sometimes I need to add that expertise. So how did I transition from flow cytometry to genomics? It just didn't happen overnight. And why did I do it? So, really the lines are blurring between flow cytometry and omics technologies. And we all know this because we have things like CytSeq and AbSeq that are coming out that, instead of using fluorescent labeled antibodies, we're using oligo labeled antibodies. And this really started kind of with CyTOF, where we kind of diverged from traditional flow cytometry by using the late, heavy metal labeled antibodies. And so it really, is important to keep your facility relevant with new technologies. And it gives you even more information. So it's, a lot of times, you can do these new things too, because you might be saying like, oh, you're gonna phase out flow cytometry, and that's not the case because all of these new technologies still need to be backed up by something that's been, tested and tried in the field, right? So a lot of the reviewers are gonna say, go back and do that experiment the regular way and show me that it actually makes sense. And so I don't think we're gonna like phase ourselves out by doing this anytime soon. So the reason why you might want to go from just flow cytometry to flow genomics is because of the droplet-based single cell methods that are out there right now. And so I'm not going to go into details on how they work. I'm happy to talk offline about all of the different techniques and the companies and how they work. We've assessed a lot of them, but the general model is to capture thousands of cells in one run instead of maybe 96, the way you do in a plate. And there are homebrew methods like DropSeq and InDrop and SQL originally. And these have merged into commercial kits like from 10x Genomics. BD has the Rhapsody. There's Mission Bio that does single cell DNA with protein. There's Parse, which is new without an instrument. There's Honeycomb Bio, which has a cute little hive that you can ship around the US. And so each of these have their niche. So there's pros and cons to everything. And so it just depends on what your researchers are doing and what makes sense to bring into your own core. But I think these have a place in a flow cytometry lab. And so if you think about where we started with single cell, it was really plate-based in the beginning because everyone was doing SmartSeq and looking at the whole transcriptome from single cells. And so when I opened the flow facility in 2016, we were sorting hundreds of plates a week, thousands of plates in a year. It was nonstop plate sorting on the Asterisk, and this is the reason why I have that big monstrous cell sorter is because the Asterisk could handle that kind of workload of plate sorting. But then I started to notice it just kept declining year after year. And so right now we hardly ever sort plates because no one really wants to do a whole transcriptome unless it's a very particular question they're asking. Everyone is doing a 3' or 5' capture, where you're looking at, actively expressed mRNA, which is fine. Like, it saves you money, right? And it's easier to do now. But because this increased, where we wanted to sort whole cells to load them into these droplet-based methods, it kind of shifted what we were doing in the lab. And so we needed to, pivot how we were offering cell sorting to researchers. And because of that, obviously when things fail, they always come back to the cell sorting because that's the easiest thing to blame. And so understanding how the protocols work and the pain points in the assays for the single cell part of it will really help you have those conversations with researchers when they do fail. So that way you can either stop them before they go on, because you can know, like, I know this is going to fail right now, or you can have those conversations when they do fail. There are certain, points in the method for single cell, especially in 10x, where if you do something incorrectly, like adding the buffers on the wrong time, then you can burst your cells open, right? So if you add the water to the cells before adding the water to the master mix, that's it. You've just ruined your whole 10x experiment, and you might not have realized it. So you might get the data back and it looks like all junk, like a lot of mitochondrial reads or something crazy. But if you know the workflow, then you can better understand how to help people too. And, lastly, like, I saw a lot less demand for cell sorting because now people are just loading directly onto 10x. There's other ways to enrich your population, so bypassing cell sorting. And so at the end of the day, it really comes down to making sure that your revenue stays up, right? Where sometimes core facilities have to generate revenue, unfortunately. Like, we don't like to. I don't like budgets. But I have to make sure that I can still cost recover in order to keep the facility going. So bringing in new technology can help with that. So there are a few things that you can leverage running a flow cytometry lab that really help on the single cell genomics side. And one of it is just cell preparation expertise. Like, we are pros at, prepping cells to go onto cell sorters. And you can use this on the 10x side or whatever single cell side you're working with, because oftentimes genomics colleagues don't have as much of this experience because we've been doing this maybe 5, 10, 15, 20 years, forever, right? And we just know all the tricks to getting cells to look good, And we have the tools in our labs, from cell counters to tissue dissociators to MACs to whatever you have, centrifuges, TC hoods, you have the whole setup. So why not use that for doing, 10x workflows? And we always say, like, garbage in, garbage out. And that's the same thing with the 10x box, right? If you put garbage in, you were going to get garbage out in your data. And yeah, my genomics colleagues are getting a taste of this. And so sometimes they come back and they're just like, man, that keeps failing and we don't know why. And then I'm like, who are you working with? And they say, and I was like, oh yeah. We see them too on the flow side, and they do not prep their samples great. And so we have the talk of, like, how to get them to do a better job. Another thing that we're really good at in flow that we can apply to single cell is training. So flow cytometry labs are known for having really robust and rigorous training programs. This is what we do. This is a big part of ISAC and Cyto too, right? We talk about this all the time. And so because we already train researchers to run samples, and it's extremely interactive and we follow up with people, you can offer this, you can pivot and offer this on the 10x side or other single cell side as well. And so that's what I do. So I do a lot of training, and I offer that, at a nice markup too. Because if you think about the relative cost of things, people will more likely pay for a 10x training than a cell sorting training, because the 10x reagents are way more expensive than the cell sorting side. And we also have really good customer service. So that's something that I like to highlight, that a lot of times when you're working with genomics cores, it's just like a drop off and you walk away and they do the work for you and give you the magical data back. But we are very much there, and we're holding people's hands and we're telling them what to do. And, we're just way more interactive with everyone. And I think that's really appreciated when people come to learn how to do this for the first time, because it isn't very straightforward. It's not an easy approach to do. And so really helping people troubleshoot those problems, and using those same skills that we have on the flow side, online scheduling tools, maintaining a lab, making sure it's nice and neat, those just go a long way. And so the ways that I got started is basically, I just talked to my genomics core and said, hey, you know, would you like to collaborate on this? Because I heard through the grapevine that they were looking for space at the main buildings because their building is a few blocks away to have a cell lounge. And I was like, wait a minute, I'm the cell lounge. Like, that's what I should be called, right? And so I reached out to them and said, hey, I heard you're looking for space. Do you want a bench in my lab to do this? People can come and drop off samples with you. And then people who don't want to do this as a service, I can train them if they want to be trained. And so that's kind of how it started. And you can ask, you know, your genomics colleagues, like, what are their pain points? What do they really need help with? Do they need help with the sample processing side of it? Do they need help with those instruments that they don't currently have in their own? So you can just figure that out and then try to fill that niche. And then you can write a proposal for the idea of your expansion to your facility. Just show like a need. Like, you know, do you have a lot of after-hour samples coming through? And you can phrase it more importantly, how many samples do you lose because you don't have this kind of situation set up in your lab? Does your genomics core do off-label work from the protocol? Like a lot of my researchers want to do weird things. So they want to like spike in their own primers. There's all sorts of stuff that RGP genomics platform won't do because they are focused on making things very high throughput that they've already tested. And then space. You obviously want to carve out a little bit of space in your lab to do this because you're working with RNA. It needs to be very clean. So we have a little bench back in the corner that we do this. And people can reserve this in our online calendar system and then also get access to the 10x boxes. And then I made friends with my head of purchasing in my institute. So he tells me when labs might be getting rid of things and I can, you know, take them in. This has saved me money in the beginning. But you can also add some things every year if you have a little bit of leftover capital equipment budget. Bioanalyzers, tape station, thermal cyclers aren't very expensive, and these are the type of things that'll help you get going. So just to finish, I'll go over how we're going to expand into the future, and then that'll be it from me. So I mentioned earlier that spatial transcriptomics is really becoming popular. And this is kind of the direction that we're going. So the 10x offers these kits for working with slide-based tissue sections. It's called Visium. They have fresh frozen and FFPE. So we've been trying to help researchers with these workflows. They're not easy. They're actually quite difficult as I'm learning. And then later this year, they're going to come out with their own imaging spatial imaging platform called Xenium, which is a box that you load your slides into, and you can get very targeted probe-based imaging, kind of like Murfish. And then we're investing more in histology equipment. You can kind of see the cryostats sitting there. This is the area of the lab that I'm hoping that I can carve out some more space and put some other tissue sectioning equipment like H&E stainers, paraffin embedding stations, and slide scanners. So that way we have a really nice workflow. If you come in, you can section your tissue, you can image it, and you can also do the quality assessment checks on those tissues to see if they're worth even sectioning on the BioA. And then, so to do more of this, we want to obviously have some like confocal-based systems as well to help with imaging. And Leica has just released a new, very easy to use imager called OMICs, just launched a couple of weeks ago in the US. We're also looking at laser capture microscopes because you want to be able to do single cell proteomics and help those groups out potentially. But they're also useful for other things. They're actually pretty cool. I just demoed it last week where it cuts out very precisely little pieces of tissue, pops it out into a tube. And then tissue processing equipment. So things like the singulator from S2, that's becoming a real pain point for us at Broad. It's like, how do you process hundreds of tissues, thousands of tissues for these really big projects? So we've tried, you know, hacking some existing equipment like the Covaris DNA shears. It hasn't worked too well, so we're looking at other equipment that's out there on the market. But, you know, all of this has been requested by my researchers. They're very appreciative to have this stuff in the lab, and so I am absolutely charging to use all of this stuff in my lab, and I'm setting up training sessions to teach people how to do all of this stuff. I don't know how to do histology. I am teaching myself, and I am bugging Kathy over at MIT, who's head of the histology core, constantly asking her questions. And so I make a lot of friends. But with that, I'm going to just say thanks to my team. I have a small team, but we do a lot. So Natthakan is he's the manager of the flow cytometry side, and AJ and Andrew work with him as the techs, and then Benoit is our staff scientist. He's the guy at the very top of the boat. He does sailing on the weekends professionally. And then Molly works with him, and Jen's our wonderful admin who does all the paperwork that I don't want to do. So that I will take questions if you have any. Also, I'm around today and tomorrow, so if you want to talk offline about anything genomics, please feel free to grab me. Okay.
Announce our next speaker, who is Simona Kola. Simona is an associate professor in the leukemia department at MD Anderson, and she obtained her PhD at the University of Modena and Reggio in Modena, Italy. Simona came to MD Anderson in 2011 after spending time at the University of Arkansas, as well as in Dana-Farber up in Boston, and has been here since, is interested in the hematopoietic progenitors and their role in myelodysplastic syndrome. She's going to speak to us today about combining flow cytometry and multi-omic technologies to dissect the molecular basis of hematopoietic disease. Welcome, Simona.
Hello, can you hear me?
Yes, we can hear you.
Sorry, I have a question. Should I upload the presentation, or you do and you change the slide?
You can share your screen.
Okay, so 1 second and I share. Okay. Could you see them? No. Possibly not yet. Okay, share screen. Could you see the slide?
We do see your presentation. We see it.
Do you see? Sorry, because I cannot hear you very well.
Yes, we can see your screen.
Okay, perfect. Okay, perfect. Okay, hello. Thank you very much for the introduction, Karen. Karen and I work together since a while. Okay, as she said, I'm an associate professor in the Department of Leukemia at MD Anderson, and I'm very interested in understanding the molecular basis of myelodysplastic syndrome and also a disorder affecting hematopoietic stem cell. This is not my usual presentation. It's a different presentation that is not going to be a whole paper discussion or a particular topic. It's just will be a mix of project that we had in the lab with the final goal of try to help you in set it up experiment that can be successful in combining flow cytometry and single cell technologies. So as you can see from my picture here, where I was looking at a Western blot result, I can understand very well Western blot result. I'm a scientist, I have a PhD, but understand biology very well. I cannot understand how to use flow cytometry, as Karen knows very well. And I do not understand how to perform multi-omic analysis. However, in the last, I would say 3 years, it's clear that these technologies together, flow cytometry plus multi-omic analysis, made my career. If I have to thank someone, I need to thank the expert at the core facility. So the first message I would like to deliver is this. You cannot do everything by yourself. Contact the expert at the flow cytometry and at the sequencing core lab and start a dual collaboration. My field is hematopoiesis. Hematopoiesis is very complicated process that starts into the bone marrow, and every disorder, at least every cancer, affect possibly the very early stem cell. Let me put the laser so at least I can show you better. Yes, this is the laser. So the very early stem cell, the hematopoietic stem cell. This is a population that is absolutely very minimal into the bone marrow. So it's very difficult to sort this population and to analyze this population. This population gives rise to progenitor cell downstream, still in the stem and progenitor compartment. Can be myeloid or lymphoid. My lab work on myeloid disease. Myelodysplastic syndrome is a myeloid disease. From the progenitor compartment, you can get all the cell into the blood and produce platelet, erythrocyte, macrophages, plasma cell, T cell, B cell. But how disease affect the stem cell is fundamental to understand how the mechanism of leukemias, of bone marrow failure. Why single cell techniques are so important in hematopoiesis? Well, it's because in the last 5 years, there's been a total revolution in hematopoiesis, particularly in the concept of hematopoiesis. And it's very clear that we cannot anymore talk about the fine population using a few antigens to dissect the stem cell, to dissect the progenitor cell. We need to talk about a continuity of cell state that you can define only if you are able to perform single cell transcriptomic or single cell epigenomic analysis. Because this cell that were previously identified with specific marker is clear that are not defined, are a continuity, are very heterogeneous. So if you want to dissect hematopoietic cancer, you need to start thinking about single cell technologies to dissect different hematopoietic state. So when you start, I'm going to be very simple and try to help the audience or who is interested to set up the best experiment in your lab. I think that when you start, I started at the beginning. I was one of the first at MD Anderson. As soon as we got the 10x genomic, the sequencing core lab, I was one of the first that started these technologies. And of course, when you are, I was assistant professor. So possibly, I don't remember exactly, in 2017, I did not have so much money. So the best for me was to try to figure it out, which is the best ratio between a good result and a good price, because you cannot spend so much to get good data. So the most important thing to remember is that first, you need to sequence enough depth and enough depth means that you need to capture enough gene into the cell. And to do so, you need to sequence at least 100,000 read per cell. Of course, if you do so, the total number and the this cost, the cost will increase a lot based on the number of cells that you are going to submit. So it's completely a ratio between these three factors. Sequencing, depth of sequencing, number of cells that you need to sequence, and overall cost. So as first conclusion, what I do every time is fundamental for me, first, to sort live cell, because that cell, apoptotic cell that you sort, will affect your result. And you pay for something that is not going to be good. First important thing, live cell. Second, contact the core facility, the sequencing, and ask for the price. I do every time. So I first contacted the core facility at the sequencing core, and I decide the price. And I ask, for 100,000 cells, 100,000 reads, how many cells should I give you to get to pay a specific amount of money? And this is fundamental because the budget for scientists is not so light. After you know the answer, then you need to work with the flow cytometry core and try to design your experiment. So in my talk today, I'm going to show some example of how I was successful in performing single-cell RNA sequencing and attack sequencing in mouse and human highly purified stem cell, as well as in human bone marrow mononuclear cell. I will first show you some example, and then I will conclude every topic with a slide in which I tell you in minimal detail what to do to get the result that I got. I think you saw this slide from Nat in my lab today when she talked about telomere. The mouse compartment, hematopoietic stem cell compartment, is defined, of course, by antigen. The stem cell is isolated based on a mix of antigen, usually are conserved over the lab, so are very well defined. And then you have all the other progenitor population. So I will not present it today to sort the hematopoietic stem cell because one of the goals was to try to dissect in the mice the transcriptome, the epigenetic, in particular way in the setting of telomere shortening in the mice, was to, of course, sort this stem cell population, but then go inside, because as I told you before, this population is a mix of cell, every one in a different differentiation state. So a most important thing is to set it up, a very clear panel in which you can clearly define a setting to sort the cell. And then this is the result. Using this setting, we were very good in sorting the real cell at the top, stem cell, you can see from here, or we were also able to sort the same cell for attack sequencing. Usually, you need to have at least 2,000 cells for group to get a good result that are able to dissect different clusters. Because, of course, if you get a few cells, you are not able to dissect different clusters. The more cells you have, and more you are able to distinguish difference. And I think that 2,000 cells for group, combining maybe different mice, can give you enough depth to dissect the different state inside the stem cell, and also to dissect the different differentiation potential of the stem cell by attack-seq. Another example similar to this, very similar. This is our last paper in Nature Medicine just published, in which we wanted to dissect the stem cell and progenitor cell population in myelodysplastic syndrome. That is a disease of the stem cell that occurring during aging. What it does is that simply patient cannot produce any more blood cell, and they die for infection, or for they are transfusion dependent, or they progress to acute myeloid leukemia and they die for progression. I'm not going to present all the study, but importantly, what I want to present is that we, together with Dr. Dewar, we analyzed 123 samples, 100, a lot, no one did this before, a sample from patients with MDS, because our goal was to dissect the stem and progenitor population in a disease such as MDS that is really heterogeneous. This means that the disease can affect platelet, erythrocyte, monocyte, myeloid cell. So if you want to understand something, you need to increase the cohort of sample. Using previously defined stem and progenitor population that are shown here, we were able to found that MDS could be divided into different subtypes. One of them was characterized inside the progenitor compartment to an increase of the common myeloid progenitor cell. This is a particular set, population of cell defined by flow cytometry antigen that should be, that one time, not anymore, was thought to be the progenitor of all the other myeloid cells, so both myeloid and erythroid and megakaryocyte. Now, after the single cell technique were established, seem not to be the same as before. But anyway, the second, and also the level of the stem cell, this particular subtype of MDS was characterized by an increase of the more primitive stem cell, the one at the top. On the contrary, the other set of sample was characterized by an increase of GMP, so granulocyte, monocyte progenitor, and increase at the level of the stem cell of lymphomyeloid prime progenitor. So bottom line, two different stem cells are expanded, suggesting that the disease possibly involve different type of differentiation state in the stem cell. We then follow up during treatment, and we characterize every of these states at the time of progression. But this is not the reason why I'm talking about this right now. I'm talking about this right now, sorry, I cannot, okay. Because, of course, as I said before, we had to validate our result using single cell technologies. Because, as I said before, flow cytometry is good, but this result need now at the level of the hematopoiesis to be validated by analyzing the transcriptional state, transcriptional differentiation state of the cell. So what we did, and these are the sample, we analyze, these are two representative samples, one CMP and one GMP pattern, so one each of the two groups that we previously identified. And we did not sort the stem cell, we just sorted the overall hematopoietic and progenitor population, all of them. But then we asked the question, so how this cluster cluster here identify the stem cell, are the stem cell between the two groups different as we observed by flow cytometry? And the answer is yes, because we were able to characterize the cell at the top, the stem cell at the top, and clearly see that the one of the pattern, the one with long-term hematopoietic stem cell expansion, that was the more primitive stem cell, indeed was characterized by expression of genes that are linked to quiescence, that are linked to primitive stem cell, like, for example, CLE, C9A, HMLT3, or MIS1. On the contrary, the other subgroup of cell was characterized by, you can see cluster 0, by the expression of genes that were already myeloid bias. So, for example, the transcriptional factor CEBPA. This transcriptional factor is the first transcriptional factor that is active during myelopoiesis, suggesting that this particular subset of sample was already prime. So the stem cells are not anymore the more primitive stem cell, but are cell downstream that possibly induce myeloid, that possibly drive myeloid differentiation. One second, because every time I have a problem to change the slide. Okay. Another paper, this is another paper, is unpublished. We are finishing the paper. Use this technique to perform single cell ataxic in the hematopoietic and progenitor compartment. This is more difficult than single cell RNA sequencing. I will tell you why later on. Here, the goal was to try to identify pattern or mechanism through which early step myelodysplastic syndrome has CCUS. CCUS is a clonal cytopenia of undetermined significance, is a pre-leukemic state. Our goal here was to try to understand how are the stem cell in pre-leukemic state, because myelodysplastic syndrome is a disease that is incurable. So our idea was, let's see before become a full disease. Can we target the disease at the early stage? So the idea was to perform a single cell RNA sequencing. I'm not going to represent, but also ATAC sequencing. You can see here we were super successful and we were able to sort hematopoietic stem and progenitor cell from healthy donor age match, because myelodysplastic syndrome is a disorder affecting aging, so the control should be age donor, aging, and also CCUS. And using this technique, we were able to demonstrate the cell in CCUS were already exit quiescence. They were prone to differentiate, and they were prone to differentiate to myeloid cell. We were very successful and in performing this result. So I think that now is the most important slide, I think, for the first part of my presentation. And I would like to share with you what we did in our lab over the year, because at the beginning we were not successful in performing a single cell RNA sequencing or ATAC sequencing. We had to set it up, the technique. How it works, the overall strategy is first you start from blood and you just do a mononuclear cell enrichment. It's clear that if you want to sort a stem cell or a particular subset of cell in the blood, you cannot just go to the flow cytometry and stain the overall bone marrow mononuclear cell, because alternatively you finish the day after to sort the cell. So you need to have an enrichment. And the first enrichment is based, particularly if you do stem cell analysis, is based on the lineage negative depletion or the CD34 positive enrichment. We use for the mice a Miltenyi cocktail that include lineage antibody, and we at the end keep the negative fraction. While for human, we enrich the cell for CD34 positive, for the CD34. So we use Miltenyi bits to pre-sort the cell. Everything needs to be, of course, in every moment on ice. This is fundamental. Then you need to stain your cell with the antibody of interest, in our case, the stem and progenitor cell, and go to the flow cytometry. Fundamental is that you cool down the instrument. You need to cool down because this is a transcriptomic analysis. Cell cannot be out at room temperature, not even for a few seconds. They continue to be at 4 degrees. And then you need to sort the cell based on what you decided with the price, as I told you before. In general, this is my experience. For single cell RNA sequencing, you should sort double number of cell than the double number of cell that you want. For example, to get 2,000 cells, you need to sort 4,000. This is because you are going to lose the cell later on in many processes you are doing. If you want to do ATAC-seq, this is more challenging because ATAC-seq, the instrument of ATAC-seq is not able to capture at the same efficiency the cell as the instrument for single cell RNA sequencing. So in my experience, I always sort 4 times more the number of cells that I sort during single cell RNA sequencing. The sorting should always be done in a collection tube that is low binding, and you should put only 4 microliter of PBS. This is what I did, pure PBS, just to a little, because the issue is that when you upload the cell into the sequencing machine, the volume is limited. So you need to decrease the volume as much as possible. And then you just centrifuge for 1 minute, and then you go to bring the sample to the flow cytometry, to the sequencing core, and you are going to be successful for sure. The second part of my talk is about the analysis of mononuclear cell that in a way seem to be more easy, but honestly is not. So for example, I start from an example of a paper that is almost finished under revision, a paper in which we wanted to understand how a mutation, a particular mutation in myelodysplastic syndrome, the mutation is a splicing factor, is SF3B1, is a peculiar mutation that is always associated to a particular type of myelodysplastic syndrome characterized by the formation of ringed sideroblast. This is a very peculiar disease, in a way that a mutation gives you a phenotype. The phenotype is ringed sideroblasts, are accumulation of erythroblasts, so cell precursor of red blood cell into the bone marrow, but these are completely abnormal. Ringed because this ring are just accumulation of iron into the mitochondria. Cells are stuck over there, they cannot differentiate, and the patient had anemia They die because they continue to have a transfusion, and then with transfusion, they have an overload of iron, a lot of complication. So the overall goal here was really to understand molecular mechanism to overcome anemia in this patient. How to do so? The idea that I had, that we had in my lab was, of course, to use new single-cell technologies and to try first to understand which is the cell that is affected during erythropoiesis. Because the production of red blood cells starts at the level of the bone marrow, at the level of the stem cell, and go through many different steps in which a cell undergo more differentiation. Starting from, I don't want to go through this, but starting from BFU that are the more primitive. And then they go differentiation until they reach the final step into the bone marrow and become orthochromatic erythroblast. This cell lose the nuclei that is phagocyted by the macrophages and then finally are released into the blood. So the issue for us was, okay, this is a disease affecting erythropoiesis. Which of the cell is affecting this disease? To do so, we perform, I cannot, okay, 1 second. We perform a single-cell RNA sequencing analysis in bone marrow. We also did single-cell RNA sequencing analysis at sick in the stem cell progenitor cell, but this is another example. Here is inside the bone marrow. So we wanted to dissect the different differentiation status of the erythroblast at the level of the bone marrow. So we analyze 5 untreated patients with MDS with this particular mutation, and we compare them with 2 different age-matched attitudinal control. Of course, the first thing that we observed was that the erythroblasts that are shown here in red were absolutely increased. This is consistent with the fact that this patient has a huge accumulation of this abnormal erythroblasts into the bone marrow. But then our goal was to really dissect the differentiation step inside the erythroblast population. So we look inside the erythroblast component, and based on the expression profile that was previously validated by Hadar, we were able to dissect all the different erythroid population. And looking at the comparison, so we compare the healthy donor with the MDS sample, we were able finally to show that MDS sample has a huge increase of orthochromatic erythroid cell at the expenses of early hematopoiesis that are the more primitive hematopoiesis. In other words, all the cells were orthochromatic cells in the bone marrow of MDS-RS. We went through, we need to validate because this now become single-cell RNA sequencing data, but now you need to have for having a good publication, you need then further validate the data using flow cytometry, using possibly different approach. And what we did was to use again a different approach, so not anymore the marker that we used to distinguish the stem cell, the orthochromatic cell by single cell RNA sequencing, but we use another panel of antigen able that was previously published and distinguish the erythroid population in 6 different populations based on the expression of CD45 that should be negative, glycophorin that should be positive, and inside CD45 negative glycophorin positive cell, looking at the BAN3 and CD4090. And we were able to show that, again, we observe an increase that is here of orthochromatic cell. Of course, then we go through this, the different pathway of this orthochromatic cell that were upregulated. We found the molecular mechanism that were significantly upregulated, and we potentially found the target to overcome aberrant differentiation, but this is another story and is not related to, I think, of your interest. So bottom line, using this technique, flow cytometry, single cell RNA sequencing, and flow cytometry again, we were able to conclude that the ringed sideroblasts were accumulated at the level of the orthochromatic cell. Another example of how to use single cell RNA sequencing combine with other technique like cytoph or again flow cytometry later on, help to dissect the cellular determinant of response to therapy. This is a follow-up paper of the Nature Medicine paper that we just published. The drug is venetoclax, so an inhibitor of BCL2. But this is not so important. The aim of this study that we are doing, this is just an example, was to try to understand not only how the MDS affects stem and progenitor cell, but how the different populations inside the bone marrow particularly are affected during therapy. Possibly trying to dissect the role of immune system or other cell, because we know that MDS is, a disease of the stem cell, but it's mostly an overall disease that affects many other cells into the bone marrow. So this is an example. We have multiple examples like this. We started using an analysis, an analysis by cytoph. Cytoph, you know what it is a flow cytometry plus protein. So we had a panel including all antigen to dissect the different hematopoietic population, as well as other antigen to dissect the pathway inside. Here I'm showing only the population. And we look at how this population change over the treatment, the therapy of this patient. And again, this is a representative experiment, but was confirmed, this result were confirmed in 5 other samples. What we found was very peculiar. We found that when patients achieve marrow complete remission, C0 is before therapy, cycle 0, before therapy. MCR mean marrow complete remission, when the patient achieve remission thanks to the therapy. And PD is progressive disease, when patient progress again. What was very peculiar was that in all 6 samples that we analyzed, at the time of marrow complete remission, we observe a very significant increase of CD4 T cell. In my opinion, this was very interesting. So I decided to go over and to perform a single cell RNA sequencing in the bone marrow mononuclear cell. And the result, these are another sample, so not the same. And we observe that indeed you can clearly see from here, this is the cluster of the CD4, and these are the identity of the cell. In blue are the cell from patient at complete remission. From the patient at complete remission, they are blue, suggesting that even single cell RNA sequencing was able to confirm that this cell that the patient at marrow complete remission had a huge increase in CD4 T cell. Single cell RNA sequencing allowed you also to try to understand which type of CD4 T cell can be expanded, because you have the profile of the cell. Based on the profile of the cell, we realize that, in our opinion, these cells were naive or stem cell memory T cell. Is this enough? It's not enough. We need to, again, to go back to the flow cytometry and to to confirm, I always have a problem, okay, to confirm the result using immunophenotype analysis of the T cell. We did not just CD4, we also did the CD8, we did the NK, but this result using this panel of of antigen or antibody that are very well established in the immunology field, were able to confirm that this cell, the cell that were expanded, were indeed a naive T cell and CD4 stem cell memory. So, and now what we are doing, of course, is go over this and try to understand the function of this cell. So this is the slide to summarize how we can get this data. The overall strategy doesn't change much, but is fundamental. This is a fundamental step that you should remember if you want to sort any type of cell. It's easy to count the cell under the microscope, and then you count the cell you bring to sequencing. Never do this, never. Because it's fundamental that you sort live cell. Cell need to be live. So you need always to go through the flow cytometry, stain the cell for a dye for viability, and then get the right number at the sorting. Again, you have to sort using a you need to cool down the instrument, and then you sort in the tube with 4 microliter of PBS. But now there is a problem here, and this problem maybe is a stupid problem, but for us was the cause of failing of some experiment. And so the issue is that when you bring the sample to the sequencing, it's very important that the volume is reduced, because the alternative, they cannot load all your sample into the machine. And it's clear that if you sort mononuclear cell, I would suggest to sort at least 20,000 cells to get 10,000 cells, because here we are sorting for many different populations. So it's not anymore a pure population, are many different populations. So you need to dissect the population very well and try to get a differential expression analysis between the group you want to compare. So at least 20,000 cells. But if you sort 20,000 cells, the volume will increase dramatically, the volume of the collection tube. We are talking about 30, 40 microliter of PBS or media. And this is not good, because you cannot bring this volume to the flow cytometry. So it's fundamental that you centrifuge the cell. But to centrifuge the cell, remember to add pure FBS in a way that your total final volume will contain 10% FBS, because you are sorting a very limited number of cells. And you cannot precipitate this cell without adding protein. Put pure FBS. And this is the reason why we were successful. At the end, we were not at the beginning. Then you centrifuge your cell, 10 minutes, 1500 RPMI, and you bring it to the core. You get your data. But again, you need to come back again to the flow, because it's not enough this for a good publication. You need to reconfirm your data, not anymore using sorting, but using flow cytometry. And one second again, why I don't, okay, this is my last slide. And this is my take-home message. I hope my presentation was different from the one that I usually do, talking about a story. But I would like to make you clear, if any scientist says in this meeting that we are scientists, we cannot do everything. And again, you need to work together with the core facility in your institution, because honestly, Dr. Karen Dewar at the flow cytometry lab and Dr. Erica Thompson at the sequencing lab are making my career. And I thanks them and thanks all the person at the core that helped me over the years. And thank you very much for your attention. I'm happy to take any question.
Thank you, Simona. That was a really nice overview of all your stuff. I think what we're going to do now, we have the roundtable session. So if we want to have Tricia and Sabrina, we've got some microphones for you down here. And then Annie, hopefully you're still on the line. We can have you join as well. And there are already some questions, but if anybody in the audience here has questions, we've got a microphone. And same thing, anybody online, please feel free to put it in the YouTube chat and we'll add them. There is not. Okay. We have some questions already that came in from the chat in the first few talks, and so I think I'll just kind of start to sling them out to you. Okay. This was during Tricia's talk, but I think if anybody else can address it, then please do. What are the most common failure points in sorting and cell prep for 10X?
I mean, there are lots of failure points. I think that's something to throw out there. But what I've seen has ranged from people just being way too hopeful for their samples, like expecting way too much from what they have, to not having enough or not planning, I think, to say in the beginning, or doing test runs. So I think it's always a good idea to not bank on your first few samples to be successful, to do a lot of pilots essentially, because there's a lot less stress, because a lot of times you have to coordinate with the person who's loading the chips. You're not doing it yourself, and you don't want to have everybody lined up, the whole team ready to go, and then you show up and your sample is absolute crap. So I think it's really important to, at least what I've seen is the people who do their due diligence and do some test runs to figure out kind of your workflow, right? How many cells are you actually going to get? I think a lot of times people think they're going to get, like the last speaker said, double, triple, quadruple what you actually need, and then they only get 2,000 cells. So I would just say plan.
And if I can add something, that is a very, very interesting question. And one thing I learned from Karen is every time you do a sorting, even if you do every day, you need your negative control, you need your beads, because something can happen at the instrument and you have a precious sample from a patient and you need to do the sorting. So if you have to do the setting again of the machine at that time, if you need the negative control, if you have a weird sample from a patient, it's not a mouse, so it changes. The immune system changes. So you need to do the setting again. And in my experience, the people in the core facility are always available if you have all the control and the right thing to sort. So that is my, and also the temperature, as Simona was saying, the temperature is really, really important. important. And we are in Texas, and in summer we have this high temperature. So if you bring your sample outside, all your cells will die. So it's really, really something important. You have the right tool to transfer the samples.
I think the controls is a very good point, because like you were saying, the patients change, and you might see a population. What you might see on the flow, plots doesn't add up, and it doesn't make sense. So having like a standardized positive control is really helpful in those instances. And so what we have is we screen a bunch of donors randomly. We bank a lot of aliquots of good blood where we get representative populations of everything, and we use those as positive controls. So that way we know the antibodies worked, right? And we know it's just that funky patient. And so then you can make the call, like, do I still sort this because it's a really rare, precious sample, and I just load 1,000 cells and get what I get? I just waste the rest of this run. Or do I, not go forward because I only have 5 cells? And so that just doesn't make sense, right? And so controls are definitely, we don't have to worry about the heat so much because I'm in Boston. So also we're in one room.
At least you don't have that
we don't have to go
problem.
outside.
Regarding the temperature, I have to comment that here the labs are at 4 degrees anyway, so here in Texas. But anyway.
Simona, you had mentioned that you spin after your sorting and then you go to 10x. Was just wondering what your take on that was, because I had done some testing in my core where we ended up we did the 4 microliters like you did, and then we just went straight to 10x. So I was wondering also any of the other people if you had comments on what the centrifuging does, helps, or takes away.
No, that is so. What I understood is that possibly I'm wrong, but at least until 2 years ago, I always understood like this, that when people load your sample into the 10x machine, the sample is limited in terms of volume. So you cannot put too much volume on the machine. Alternatively, the problem is this: when you sort small amount of cell, like stem cell, 2,000, 4,000 cell, well, the amount of liquid, in a poor word, okay, I'm very bad at explaining with a technical word, in very simple word. So you sort the cell, and then you get the cell in some liquid from the flow cytometry, right? So if the number of cells you sort is very low, this liquid is going to be 5 microliter. That is good. But if you are sorting something like 20, 30,000 cells, this liquid will be 30, 40 microliter. And then the problem is that if the volume that can be uploaded to do single-cell RNA sequencing is limited, the personal single-cell RNA sequencing is going to use the total volume allowed for them and then discard all the rest. And so at the end, instead of sequencing the number of cells you wanted, you sequence a little amount of cell. So to avoid this problem, I centrifuge the cell if the amount of liquid, the volume that I collected is more than 30 microliter. And to do so, I add FBS, pure FBS, because you need to precipitate the cell.
So I noticed in your talk, Simona, that you had a picture of the 10x Connect, which is the automated 10x system, the robot. So I'm not sure if your genomics core is now using that, but the input volume for the Connect is actually 10 microliters. And so that might be where that number is coming from, because you load your samples in a 96-well plate on the Connect, and it makes everything for you from the GEMs to the libraries. But the maximum volume you can load on the Connect is 10 microliters.
Exactly. That is why I sort the cell.
Yep. And then I can follow up on that and say that we do a lot of nuclei.
I centrifuge the cell. I not sort. I centrifuge the cell. I need to centrifuge the cell because alternatively they will never upload the total volume, and I lose I lose 1/3 of the cell because I got 30 microliter. Exactly. This is fundamental, and that was the reason why at the beginning I failed my experiment without knowing. It's very stupid, and I'm sorry, I'm not a flow cytometry person, and possibly it's very stupid, but at least I want to share with you because in my opinion it's very important. If you are a scientist, you don't know this thing.
I've heard like way more failures from way more stupid things. So I think I can give you a long list of things that have failed at Broad.
Every experiment is $20,000, $30,000. So.
Oh no, we've wasted many hundreds of thousands of dollars. But for nuclei, so single cell nuc-seq, right? That's a very popular. And a lot of researchers don't want to spin that down. So what do you do? So what we do is we sort into PCR strip tubes, and we sort into almost no buffer, so like a microliter or two, and directly into that tube. And then your concentration with this, and also exactly, and with the 70 micron tip on the Sony or the chip, and so it's very limited buffer. And so that way we don't, and this is just regular chromium, not on the Connect. So you still get like 20, 30 microliters, but it's much smaller and, yeah. So there are ways to get around it. But yeah, I have these arguments back and forth where I tell researchers you need a centrifuge and they're like, no, I refuse. And so we have to figure out a way to do that. And so usually if we can, we run it on a smaller nozzle size.
Okay, so this is a question for Tricia, but we also have questions for all the other panelists. So Tricia, when you were integrating your flow cytometry with the genomics, how do you address the data analysis on the genomics side?
That's a good question. I don't do it.
You outsource it with another core? Or?
Yeah, so if you're doing sequencing through Broad, then Broad has bioinformaticists like embedded within the genomics core. So we have a whole platform that only does data analysis. But the other side of that is a lot of labs have their own bioinformaticists at Broad. And so each lab will have 1 to 2 people who just do data analysis for the groups. And so I actually tap into them as well because I don't know how to do, I get my sequencing data. I'm like, okay, what's the next step? And so I reach out to my, team. But this is something that is on my wish list to learn how to do my own data analysis. But yeah, it's a skill. Doing single cell RNA-seq analysis is really difficult. So but I have to say there are companies coming online that will offer this as a service. And so we're kind of looking into them because you can download. Almost like you have access to FlowJo, right? Or I guess, there's SeqGeek too within FlowJo. But there are companies where you can just send them your data, your sequencing files, and then do all of that analysis.
Probably faster.
Faster, like you can charge for that too. You can build that into your cost model, right?
Yeah, so the next question is from Deanna from Peter. This is also to Tricia. Can you comment on the genomics impact of cellular debris from cell sorting? So do samples for sorting with a lot of debris give bad genomic results, even when you try to sort single cells?
Yes. I think the short answer is like yes. Yeah, so and this is a big problem with multiome. So multiome is doing attack and a single cell RNA-seq. So you're having to go after nuclei and whole cell. And a lot of the tech dev on this is dissociating nuclei from tissues that looks good. And so there's just many iterative rounds of dissociating tissue, looking at them under microscopes, seeing like how much debris to nuclei do I have, and is that going to work? And a lot of times, it has to be really clean preps. And so you just have to optimize your protocols. There's a lot of different methods for isolating nuclei, down homogenizers, ultracentrifugation. The 10X has a protocol. It's literally called the Frankenstein protocol. And you have to tweak it for different types of tissues.
Right. Do you use cell tracer dyes to pull out when you sort for sequencing to pull out the cells from the debris?
Yeah, I always recommend using multiple dyes if you can. So
Right.
live and a dead cell stain, right? So something DNA tagging and something
Right.
live, like, calcine, if you can, if you have enough room in your panel to be able to do that. That way you can only target the cells that are really healthy.
Anyone else want to comment?
No, I totally agree with her. I mean, it's fundamental, as I said before, to sort a live cell. It's fundamental. Alternatively, you will lose money in sorting something that is not going to give you high quality data, because you will find a cluster full of mitochondria gene and you need to discard them. So it's fundamental. Never, never, never go straight from a microscope counting cell to the sequencing. You always need to go through the sorting machine for a live cell. This is my personal experience. In my opinion, it's fundamental.
Mm-hmm. Thank you. Yeah, so we have another question from Diana to Annie. So first, Diana says, excellent talk, Annie. So for the infinity flow, how do you proceed with antibody titration, and how are controls incorporated in defining positivity for a certain marker? And the other question, for the infinity flow, how do you proceed with antibody titration, and how are controls incorporated in defining positivity for a certain marker?
Yeah, those are excellent questions. So unfortunately, for the infinity flow, you have to titrate one by one, like starting with 1 to 25, 1 to 50, all the way up to 1 to 800. So for us, we focus on c-KIT positive cells. So we enrich with Miltenyi, and then we titrate like 100,000, 200,000 cells per concentration for each antibody. So unfortunately, there is no way around that. And I do not recommend anyone trying to attempt this by, just throwing antibodies on. It's not going to work. In terms of gating for positivity, like positive versus negative population,
right.
I'm imagining you're asking me about this in the inferred matrix, not the real life flow. So this is a complicated question to answer, because for me, I'm not just interested in what's positive and negative. I'm also interested in the definition of high, medium, and low, especially when it comes to reporters. So when it comes to that, the way I normally deal with it is, first of all, for reporters, I like running them on the spectral because it gives me better resolution. So you have experience from real life flow outside an infinity on how they should look, right? And for the infinity markers, the things I put on PE. Now, when I look at that inferred FCS file, it's worth noting that the way we have extrapolated this, the interpretation of the antibodies from sequencing to the antibody signal from flow, is a very complicated machine learning algorithm where we correct for the range and dynamic range of expression of each antibody. That's how we know where we should be putting the gate. And of course, everything you gain from the infinity flow needs to be validated with real life flow before you try to do anything else. So for example, you say there are these two markers, I gate for like CD11B high versus, like LY6C medium, something like that. Now, then you have to design a real flow panel with both of those markers on an actual fluorochrome that can be separated and see what they look like in real life. And then you sort for them for validation. So before you have the validation step, everything is a hypothesis.
Thank you. The other question also for you from Sarah is, how many cells have you targeted in your site-seq? And how many samples could you stain with one vial of total seq? And the final question is, have you included any exclusion threshold for ADTs at analysis?
Yeah. So the first question is, how many cells have I targeted?
Right.
So in my atlas, I have over 190,000 good quality single cells. And we're talking about 1% of KIT positive cell populations, down to 0.001% of total KIT positive populations. And I have really good resolution of every single cluster. So overall, I think this is from 160 C57 black 6 mice, a lot of sorting, a lot of sequencing. And then in terms of like how many number have I targeted per experiment, Now, I've managed to do sequencing on less than 25,000 cells, and that's really difficult. So you have to watch out for the concentration of the staining, not just the volume. Like they both are important, but you have to prioritize the concentration first. And then in terms of the, I'm sorry, what's the next question?
So let me see where it is. Right, have you included any exclusion? Right. Threshold.
Oh, and then, yeah, so per vial, the way we design these lyophilized vials with BioLegend is we targeted 500,000 cells per vial. But I mean, of course, if you're doing custom reagents, you can negotiate that with the company. And then the threshold for detection. Yes, we have a very stringent background exclusion algorithm that I have designed and coded. So unfortunately, I cannot disclose that right now, but you will be able to have access to my pipeline once the paper is out, hopefully soon.
Good, thanks. So we have actually another question to you, Annie, but again, if anyone has also any comments, I guess these are more specific to Annie's work. But Svetlana asks, so when you're using the Curex system or unit to perform the laminar flow washes of the samples with antibody titrations. First question, how many washing cycles did you perform?
We did 25 cycles with a flow rate of 10.
And how many cells did you start it with?
I have done anywhere between 10,000 upwards to 1 million per well. I will say that I think you guys probably have a Curex rep over there. So as you go up in the cell number, the cell loss actually goes up. And it makes sense because you're letting the cells settle naturally onto the bottom. So the more cells you have, the thicker they will be. So as you imagine when the liquid flows on top, right? If the cell layer is super thick, you're going to lose, some, a little bit more percentage wise. But in terms of single cell sequencing, now if you're doing the typical Chromium like ChIP-G, the max loading is usually around 16,000 to recover 10,000 cells. If you're doing HT, of course, the loading is a lot bigger. But normally nobody really loads 1 million cells into a 10x port. So you just have to consider, do we really need that many cells to start with?
Right. So I guess you answered the last question, which was, have you assessed the cell recovery after the Curex washes?
Yeah. So after the wash, you are left with 25 microliters in each well. So normally for something under 50,000 or so, the recovery is like above 80%. As you go up, you lose a little bit more. And my experience is limited with murine progenitor cells, which are not super sticky or adhesive. So I think if you're dealing with different cell types, you'll have to do the testing yourself.
Okay, thank you.
Mm-hmm.
Anyone else has any other questions for the panelists?
I think I have a question for Annie as well.
Mm-hmm.
So the validation you did with the CytSeq antibodies was really amazing. I wish more researchers would do that. How long did it take you to do that?
The validation of the CytSeq?
Yeah, the titration.
Oh, the titration. Oh, gosh. I mean, the design of the experiment, also working with BioLegend's team, which was a very smooth experience, was a long time because really to be like all of that had to be done in one day. So it was basically 10 hours of sorting, plus harvesting in the morning and staining and sequencing afterwards. So I think the experiment took roughly a little over 20 hours straight, with the help from everybody in the lab. But it was very rewarding because we did nail it in one shot. So we are hoping that, this is like something like a new concept we're bring to the field. And in fact, if, with our biological discovery being published, we hope that this cocktail can be applied to many of our colleagues working on the same populations.
No, it's great. I hope more people take advantage of the work that you've already done.
Thank you.
Absolutely. Should be part of some guidelines.
Yeah, I mean, we don't really, it's like the Wild West right now, right?
Right.
So,
exactly. Yeah. Okay. If nobody else has any questions, I want to thank the panelists and all the speakers. And thanks everyone for participating.
All right, I do have some little business before we go to, hopefully, we have some of our our invited emerging scientist here still. So if you would, if you are here, if you could come down, we do have. It's been a long tradition of FlowTex after we started making, well, not super long, but long enough where we got to the point where we stopped doing posters and we decided to highlight our emerging local scientists in a different way, which is with bringing them to FlowTex and having them give small talks. But another thing that we did is we've inverted it. We don't give honorariums to our speakers, our major invited speakers, but we do give small honorariums to our emerging scientists. So if I could get our emerging scientists that spoke today to come down, I'd like to give them their honorarium, and we give $200 to each person, so that it's a small bit, but you know, $200 to someone who makes six figures isn't much, but $200 to grad students and postdocs is a little bit more. So we want to show that they're appreciated. So if we have either Sophia, Katharina, Cheng-Yen, or Natthakan, and I'm sorry if I'm or Sarah, you could come and grab your check. I'd appreciate it. Do it. Hey. All right. Sarah. Oh, I don't know, but this just goes right back to the flow core anyway. No. Oh, I'm sorry. It ran away. There you go. And I think that's all I think we lost our other two temporarily, but don't worry, they will get it. We'll figure it out somehow. Thank you so much. Thank you. And thank you everyone for joining us today. We are going to go to all the viewers out and about. We are going to now go and partake in our ice cream social, which I've also been told, besides chocolate and vanilla ice cream, we have Guinness flavored ice cream. So y'all are missing out for being not in person, but I appreciate you all coming anyways. Please join us again tomorrow for our last day of FlowTex, and hope to see you then.